and it allows us to dynamically simulate the biochemical processes within a marine ecosystem. So what I'm talking about specifically is food web interactions. And in Atlantis, this is done by tracking a common currency among all organisms. The nutrient waste flows are tracked as nitrogen and silica. And here we can also account for important ecological processes, so things like migration and natural mortality events. Simulations in Atlantis simultaneously run an oceanographic submodel, where here we can account for the climate and the hydrology of the marine ecosystem. So things like currents moving into and out of the bays, as well as the upwelling and downwelling of nutrients throughout the water column. Data is further integrated in Atlantis in the exploitation submodel, where here we can account for the human-induced impact on the marine ecosystem. So things like fishing, tourism, coastal development, energy use are all accounted for here. And at the top of all this, we can use Atlantis as a formal assessment model in this sort of policy gaming tool that's built in, where we can simulate in our virtual marine ecosystem a policy of our choosing, and then, excuse me, and then assess the impact of that policy within our virtual system before going out and implementing the policy in the real world. So you can see how all these dynamics are working together at model run in order to give you the most robust representation of the marine ecosystem. Another really exciting feature of Atlantis, and one that sets it apart from other ecosystem modeling platforms, is that Atlantis is spatially explicit in 3 dimensions. So using these yellow polygon structures, we can actually bioregionalize the Tasman and Golden Bays into smaller subregions that allow us to separate out things like shallow versus deep water habitat, as well as different benthic bottom types, like a rocky versus a sandy bottom type. And really importantly here, this allows us to spatially and temporally separate predators from their available prey, as well as fishermen from their available catch. So I showed you what Atlantis is. Now I'm going to show you why we want to use Atlantis in Tasman and Golden Bay. So just for a bit of geographic reference, Tasman and Golden Bays located at the north end of the South Island, as shown here. And this is really an economically important region for New Zealand. And that's because regionally this has a large seafood and fishing industry. It's also home to the world's only alpine parrot, the key. And it's really important for recreation and tourism. Since I've come to New Zealand, this is one of the places that everyone recommends to go and visit, and so we really want to preserve the beauty of it for future generations. And with Port Nelson being one of the larger ports in the area, it's also very important for energy and transportation. Another reason that we want to use Atlantis is that this is an ecologically important marine ecosystem. So this is a highly productive marine ecosystem. If you can see from the figure here, there's a lot of marine organisms that call Tasman and Golden Bay home throughout various life stages. Also, the bays act as a buffer to the open ocean, filtering out nutrients and sediments that run off from the rivers into the bays during storm events, and also protecting the coasts from heavy winds and heavy wave events. And then there's also home to critical juvenile habitat for things like black swans, blue cod, fur seal, and juvenile snapper. And the shallow waters of the bays act as an important habitat for shellfish, such as the scallop shown in the figure here. So I showed you what Atlantis is and why we want to use it. Now I'm going to show you how we've configured the Tasman and Golden Bay Atlantis model. So Tasman and Golden Bay Atlantis consists of 51 model functional groups. If you're unfamiliar with what a functional group is, it's just an aggregate group of species that shares some sort of similar diet, habitat, or niche in the marine ecosystem. Of the 51 groups, we're representing 171 single species in Tasman and Golden Bay. This includes bacteria all the way up to seals in this ecosystem. 21 of the 51 groups are managed or assessed stocks, and these are modeled as single species groups. And then 28 of them are harvested, including both fish and invertebrates. The fishing industry is accounted for through the inclusion of 12 commercial recreational fishing fleets. This includes trawl, line, purse seine, set net, pots, jigging, diving, trolling, seine, and dredging. To estimate the abundance of each of those functional groups within the Tasman and Golden Bay Atlantis model, and to separate that out by polygon, we used NIWA trawl surveys, which were conducted between 1992 and 2013. And we took the average over this time to give us a measure of relative abundance, which we then scaled by a catchability constant to give us a measure of absolute abundance in 2002. We then plugged that into a stock assessment model, and then we back calculate to a pre-fishing level of abundance, which was equivalent to biomass or abundance in 1900, and this is what we used to initialize the model. And in order to get that by polygon, we scale that by the catch rate from the trawl surveys. So species in Atlantis are constrained to particular habitat types, just like in a real marine ecosystem. So you can see we've assigned 3 different benthic bottom types in the Tasman and Golden Bay Atlantis model. This includes flat, shown on the left, rocky in the middle, and soft on the right. And the proportion of each is shown in the scale at the bottom there. So important to note here is that you can have overlapping bottom types within any particular polygon. So at the bottom you can see that there's about 80% flat, which is representing a sandy, shelly benthic bottom type. And then in the middle there, rocky, there's about 10% in those bottom polygons, which are going to be like rocky outcroppings. And soft is representing like a muddy bottom type, and this is shown in the one on the left there at about 10%. The oceanographic data in the model couples a full year of ROMS outputs, which is then recycled across years. And this allows us to reflect the seasonal and diurnal variability in things like salinity, temperature, nutrient advection, and currents moving into and out of the bays. In Atlantis, the species are linked by what they're eating and by who's eating who. And so you can see that this complex food web diagram is not so that you can squint and try to figure out what it's saying, but just to show you that it is quite complex. And just like a spider web, for example, you can think that if you knock off one of these little webs connecting the species, that you could end up seeing the whole thing collapse. And another really important point here is that species in Atlantis can starve if their prey source becomes limited, and they can also increase in abundance should prey availability increase and their predation pressure decrease. So accurately representing these linkages is really important if we hope to achieve realistic model predictions. And so in order to do this, we've used NIWA gut content analysis data from our NIWA scientists here. And we've incorporated that into our input for our model. So I showed you what Atlantis is and why we want to use it. I've also showed you how we've configured the Tasman and Golden Bay Atlantis model. Now I'm going to show you where we're at with the Atlantis model development to date. So when I started here at NIWA about a year ago, I was given the model as it was built, so all the data inputs were already there. And it was my job to tune it and calibrate it and get it usable, essentially. And so the first step in this is calibration of the Atlantis model. And the first step in calibrating your model is ensuring that the model can run indefinitely without crashing. And so you're not really looking at dynamics of species at this point. You're just trying to ensure that the model doesn't crash. So it can run for a couple hundred years of simulation years. And once you get past that, you can go on to tuning your model. So now you're tuning your functional groups in your unfished version of the model, which is considered here your base model. And you want to tune these groups until they reach a stable equilibrium state. And I'll explain what that means in the next slide. And then once you have that tuned, you can start turning on your historical catch removals. This is done by forcing a time series of catch removals. We have catch information for Tasman and Golden Bay from 1900 to 2012, and so we've incorporated this into our model. And then when you turn that on, you might have to do some further tuning because groups can crash at this stage during periods of heavy fishing. And the next step that you move into after you've calibrated your model is validating the model. And the main takeaway from this is that you want to match your observed biomass, so what you're seeing out there in the real world from NIWA trawl surveys in this case, to your model predicted. Once you have your model validated, you can consider it ready for use. So back when I started, the model looked something like this. And this is, again, the unfished version of the model. So the only thing that you have going on here really is your ecological interactions, your species interactions. So you'd expect that you could get these groups to stabilize, meaning that there's not too much predation pressure on them and there's enough food that they can survive. So what I wasn't looking for was all of the red arrows, where you can see these big increases and decreases in biomass, basically unstable dynamics. What I was looking for is the green thumb here, which is, in this case, showing the mussels group, the biomass over the simulation period, where the biomass has stabilized, and so that was the goal. So after several months of tuning, I was able to improve most of these groups, where you can see the green arrows now, and still this is the unfished version of the model, where most of the groups have reached that stable equilibrium state. And then, like I mentioned, once you have that done, you can move on to turning on your historical catch removals. So now this is the fished version of the model. So the only difference between the two models is that here fishing is turned on. And so you can see that these changes in abundance are caused by catch removals alone in the model. And like I mentioned, some further tuning may be needed here if a group crashes or goes extinct under a period of heavy fishing. And heavy fishing historically occurred in Tasman and Golden Bays around the late 70s, early 80s. So you can see some of the groups declined, but then when fishing pressure was reduced, the groups increased. After you've tuned your model and it seems to make some sense, you can move on to the validation process. And this is where you're comparing your observed biomass, which is shown in these figures here, as the dots, to the model predicted, which is shown as the line. So here I'm just showing 3 of the 18 groups that we had observational data for. It's the biomass over the simulation period from 1993 to 2012. And it's for just 3 groups: barracuda, leather jacket, and tarakihi. And you can see that the model is doing a pretty good job of matching these observed observational data points. Another thing that we really wanted to focus on with this was scallops. Scallops has, in recent years, crashed and the fishery has closed, so it's been a group of concern. So looking more closely at the scallop trajectory in the Atlantis model, here I'm showing the historical catch removals from 1993 to 2012 by the gray bars. And still the observed biomass is shown by the black dots, with the model predicted being shown by the black trends. The blue line there is showing you what the estimated abundance of this group was in 1900. So what you can see is that the early part of this time series, Atlantis is saying that when catches were high for scallops, that the biomass of scallops declined, which is good. This is what we wanted to see. And then when catches were low, scallops recovered. And you can see that around 1998 to 2002, that the observed and predicted are doing a pretty good job of matching one another. However, you'll notice in this period, from 2003 onwards, that the observed and the predicted start to diverge. So the observed is suggesting that the current population of scallops has declined and perhaps continues to decline in recent years, whereas the model is predicting that under this period of low catches, which we know this is when the fishery closed, that the scallop population should have recovered. And so remember that this is the fished version of the model, where the only thing that we've turned on is catch removals. So this is suggesting that something else is going on in the ecosystem that we're not capturing yet within the simulation. So one suggestion or idea was to test whether or not The possibility for an underestimate of catches during this period was possible in order to better match the observed to the predicted trend. And so in testing this, in 2002 to 2012, I scaled the catches by 2 to 10 times. So catches were really low in some of these years in order to try to better match the observed to the predicted trend in this period. However, you can see that Atlantis is still not doing a good job of matching the observed trend. And that's because within Atlantis, we're using a Beverton and Holt recruitment relationship, which this recruitment relationship says that when spawning stock biomass gets really low, you'll have the highest number of recruits coming in. And so this is what's happening here in our simulation. You can see that around 2007, the scallop abundance is really low. So in the subsequent year, you get that really big spike in recruits coming in. And then due to the fact that there's not very many adults in the system, they get fished down in the subsequent year. And again, you get that spike coming in the next year. So we're still missing something here within our simulation. And it doesn't appear that catches alone are driving the current abundance that we're seeing in scallops in the Tasman and Golden Bays. It's possible, based on what we're seeing out in the environment, that it's something to do with habitat degradation, perhaps increased turbidity in the system, making the habitat that used to be preferable to the scallop recruits, they're not able to settle there anymore and grow. So we're going to start incorporating new data sets in order to better represent this as we move forward. For next steps, I've currently submitted a validation paper to a peer-reviewed journal. And this is an important part of validation as well, because we want to get our work out there and have experts in the field review our model and make sure that it is sound, it does make sense. And then from there, I'm currently about to start running some preliminary management scenarios with the model as it is. And these will be based on the stakeholder feedback that we've gotten from the Sustainable Seas meetings. And then moving forward, I just want to note that Atlantis is an evolvable tool, excuse me, where you can keep adding new data as it comes in. So there's several ongoing Sustainable Seas projects. And as this new data comes in, we're going to update the model. We're going to improve our simulations to better represent reality. And from there, run new and improved management scenarios. So that concludes my presentation. I appreciate you all listening in. And thank you. Let me know if you have any questions.
Okay, thank you, Michelle, very much, particularly as she's got a horrendous cold, and we really appreciate you coming in. So if you have a question in anywhere in one of the rooms, then please use the raise your hand button, and we will see it here on the chat or on your icon, and then we can unmute you and you can ask it. So if you have a question, please do that. In the meantime, does anyone here in Wellington have a question to ask? Adette, you might need to come down closer to the microphone.
Is this close enough?
I think so. Can people hear her?
Nice work, Michelle. Do you have a feel for how the spatial distribution of the scallops might be affecting their dynamics, how they're responding to the fishing?
Yeah, so part of the validation process is just to look at the ecosystem as a whole. At least that's how the models have been validated in other systems. So spatially, I haven't really delved into what the scallops are doing, but that's definitely on the table for something that we need to look at.
Okay, so I can see some little video screens. If anyone in one of the other physical rooms has a question, if you raise your hand, I will unmute you. Who can I see? No, not
I
that
have
way.
a question in the meantime.
Okay, question from Craig. Come and speak
Probably
up.
shout. Can you talk us through the tuning process? So, you started off with things looking dreadful, and so what did you go through to?
Yeah, good question. So for tuning, things that you're adjusting are like growth of the individual or the functional group, as well as the recruitment, so eggs coming into the system. particularly Beverden Holt alpha parameter. There's also, so if a group is exploding or increasing a lot in biomass, you can stabilize it by increasing the other mortality term that's applied to it. So stuff like that, just adjusting it. It's just a sort of trial and error.
So did you work through looking at the worst ones, tune them?
Yeah.
Yeah, okay.
Yeah, exactly. So if you see one that's going crazy, then those are the ones you focus on, definitely. And then the minor tweaks you sort of save for the last.
Okay. So did anyone else? We've got a question coming through on the chat, which is from Andrew Baxter at DOC, which says, how do you deal with confounding pressures such as sedimentation from land and dredging, and then resuspending and sorting sediments?
Okay, that's a good question. Thank you, Andrew. So right now we don't have any sedimentation plumes or anything that's occurring in Atlantis. Like I mentioned, we went from an unfished version of the model to the fish. So we start off with our initial conditions and then run the model forward in time. If we wanted to realistically account for the variability in sediment and stuff like that, we would have to actually force a time series of that data into the model. And so once we get that data, we can start incorporating those kinds of things and answering that type of question.
Okay, thank you. We've probably got time for one more if anyone has one. We have one at the back.
But only if I don't want to stop anyone else.
No, I think you're good. Come on down.
Okay. Yeah, sure thing. Okay, yeah, thanks very much, Michelle. I was just wondering if you just explain a little bit why Atlantis caused these big annual kind of jump ups and downs when you were trying to tune it, to do with recruitment.
Yeah, so that was, so the question was about the plot where I was increasing catch, and recall that during that time the abundance of those groups were really low. So this has to do with the Beverden Holt recruitment relationship, which is slightly complicated, and I don't want to get into much detail about it, but essentially it has to do with carrying capacity in the system. So when the stock is really low, they produce a lot of eggs, and you get a big spike in the abundance in the subsequent year or subsequent time step, I guess.
Okay, thank you.
And someone at Nelson was giving quite a vigorous wave, so hopefully this technology has worked and you're unmuted. Go, Nelson.
Yeah, so we just had a question about going forward. You know, it seems to me that the testing of the model is around fisheries management primarily. And are you going to have an opportunity to I guess, test another management issue that we have in Tasman and Golden Bay, or is it really just going to be looking at the fishery management question or issue to test the model?
So yes to both of those. We are currently, so the way that the model was built was it was built around a fisheries-centric idea. So all validation papers on it, which is what I've used to validate this model, looking at other papers, are sort of fisheries-centric. So you use your known data from fisheries in order to validate the model. So that's sort of why it's fisheries-centric at this point. However, Atlantis can be used to do all sorts of things. And once we move forward from here, you know, the main thing was getting it validated so that it seems to represent reality. And then, yeah, we can start addressing those other questions. And I think the focus on scallops was due to the fact that that stock had collapsed, and there was a lot of political background there that we wanted to focus on it to sort of address it and get that answered.
Sure.
Well, thank you very much, Michelle. I hope you're feeling better soon, and that was really interesting. So just if everyone could show their appreciation one more time.